And we'll break them down accordingly to, like I said, how they're transmitted. Now, these are all chemical signals that are going to have to bind to a receptor. What category we put them in is not based upon what the chemical signal is. It is how is it transported to its target. Okay. Meaning that the same chemical can be in all four classes. It depends on how does it get to its target. If it gets to its target in blood plasma, it's called a hormone. So they work over long distances, right? So the reason, they don't have to be long distances, but they can travel over long distances. So you're going to have a gland or a cell that's going to secrete the hormone, it's going to get absorbed by a capillary, and that capillary then, the blood in that capillary is going to be transported to everywhere in the body. And the only cell, every cell is going to receive the hormone, but the only cell that's going to respond to it are the ones that express the receptors to that hormone. That kind of makes sense? Paracrines work local. The word para means adjacent to. So these are chemicals that are secreted by one cell here, let's say cell A, and there's another cell over here, cell B. Cell A secretes a paracrine. Cell B has a receptor. It binds. Cell B gives me a response. These could be right next to each other. Right? This is the interstitial fluid. Right? So it's going through ECF, just diffusion, right? Simple diffusion. Now, because diffusion takes longer over greater distances, this is not going to have an effect on a cell way over here, because it would take forever for that to get there. By the time it got there, it'd be so dilute, nothing would happen. Right? So these have to work over short distances. Now, some examples of paracrines, the same chemical could also be a hormone. So, for example, epinephrine is a hormone, but it also functions as a paracrine. There's a closely related a compound to epinephrine called, which is adrenaline, called norepinephrine. Just a little similar, a little difference in structure. It's a neurotransmitter. Now, what makes a paracrine a neurotransmitter is if cell A, if this was a neuron, right, that would make this a neurotransmitter. So all neurotransmitters are paracrines, right? They're paracrines that are secreted by neurons. That's what makes it a neurotransmitter. So dopamine. Dopamine's a paracrine. It's found in the central nervous system, and it is released by neurons, and it affects other neurons. Okay? But it's still a paracrine. It's not a hormone because it's not transported to its target in the blood. In fact, it doesn't cross the blood-brain barrier. It stays exactly within the brain. It can't be absorbed. Sometimes a cell will release something like this, and it will also have a receptor for that. In this case, this cell is regulating itself. Right? Who's it communicating with? Not somebody adjacent, but itself. Right? So this case here, this substance here, is now called an autocrine because it's self, it affects the cell. So they're just terms. Like again, but I don't want you to think that that means chemistry. It doesn't make what it is, it's just how it's transported. If its target is a long distance away and it's carried by blood plasma, we call it a hormone. If its target's right next door and is carried by diffusion through interstitial fluid, it's called a paracrine. If the cell that released that paracrine is a neuron, we call it a neurotransmitter. If the thing that's being released by this cell also affects itself, it could affect something else as well. It could also affect that cell over there. But if it affects itself, it is also called an autocrine. Right? So it's just some terminology of how we label these different things. What they all have in common is that the targets are determined because there is a receptor specific for that signal. Okay? So insulin causes cells to increase the uptake of glucose, right? We mentioned it causes them to put in more glucose transporters so they increase the uptake of glucose. Well, the way that works is because there's a receptor that's specific for insulin on the surface of those cells. If the receptor's not there, or there's less of the receptor, it's not going to work as well. Does that kind of make sense? So we have a few ways to modulate this information flow with chemicals. You can do it by altering the amount of the signal, right? You can do it by changing the amount of receptors, or you can do it by altering the what? The binding site, and that affects what? Affinity. That affects the affinity. Right? So you can change the number of signal, change the amount of receptors, alter the affinity. All of those are things that would modulate the response, right? More signal, more response. More receptors, more response. Higher affinity, more response.